I'm Corina Nagy, and I am a researcher at the Douglas, also part of McGill University and the Ludmer Centre. My research focuses on the gene programs that change as a function of sex in the context of various psychopathologies. I use single-cell technology to zoom in on these changes, which include both looking at the cell in suspension, but also at the cell within its proper environment in the brain. Spatial transcriptomics allows us to look at all of the genes that are expressed within a given cell, but it also allows us to maintain where that cell is within its environment in the brain. The brain is extremely well delineated and it has many different regions, but even within those regions, there are layers and have a particular organization. And we know something about those organizations, but we don't know how the cells interact within those layers, within those regions, and how they are influencing their immediate neighborhoods. This new technology is giving us further insights into these interactions between cells in a given brain region. We can then extend that knowledge to how things change in a pathologic state. A lot of neurological disorders have gross anatomical changes. For example, if you look at Alzheimer's, you'll see plaques and tangles and actual changes within the brain structure. In depression, these changes tend to be a lot more subtle. Gene regulatory programs are changing, but it's happening in specific cell types. And in addition to that, it's happening in a sex-specific manner. The manifestation of depression can have different types of symptoms in males and females. For example, males will have more impulsivity and aggressivity and substance abuse disorder, whereas women will present with more anxiety. It's possible that we have different roads that are getting us to these endpoints. And this is what the biology is showing us more and more. Specific cell types seem to be more dysregulated in females, whereas other cell types are more dysregulated in males at the transcriptomic level. And what we saw is that in females, there was an overwhelming abundance of changes in microglia. So microglia are the immune cells of the brain. Females have a larger prevalence of autoimmune diseases. So it was quite interesting to see these immune cells of the brain pop up as particularly relevant in the female manifestation of depression. In the males, we were seeing primarily excitatory neurons in the deep layers that were being changed. So one of the things that we're trying to do now is look at the protein profiles on a given cell. Then once we have captured that information, we can actually look at what the transcriptome of that particular cell type is. So for example, different cells in the brain can be expressing the estrogen receptors. So of the ones that do, how does their transcriptome change? What does the neighborhood around cells that express estrogen receptors look like? A lot of my work is really looking at these sex-specific milieus in the brain and how they're influencing our individual cells. Because the Douglas Brain Bank has extremely good characterization of the brain, we have some of this data. So we could, for example, have some information about whether or not the individual presented with more insomnia or hypersomnia, there was a substantial weight gain or weight loss. The other way we can test this is work with animal models and try to induce these phenotypes. But it's not straightforward. For example, you can induce some of the metabolic dysfunction, but a lot of these are really human symptoms. But we have to work with both because postmortem brain tissue has many limitations. Confounds, the fact that we can't then test hypotheses directly in that tissue. There's no way to see how altering that one gene would change the physiology of the whole brain because we can't do those manipulations in postmortem brain tissue. It's a challenge, but every small incremental finding we have is giving us more information about how we can move forward. One of the most efficient adjunctive therapies to depression is exercise. It's also preventative. There's been a lot of research showing that any bit of exercise leads to more days of good mental health. So we're doing some exercise research to see whether or not the beneficial effects of exercise can be mediated through extracellular vesicles. EVs became this really interesting biomarker possibility that could give you mechanistic insight. Because EVs come from all cell types, which also means that they're coming from brain cells. And some of the early research that we did, we were pulling EVs of neuronal cell types out of blood. So not only are we finding a biomarker, but we're finding a biomarker that has mechanistic impacts. And we want to know how or where this is imparting its influence. We need to kind of go into the single cell stuff. EVs have this nice profile on the surface of their membrane, and a lot of those are signaling molecules that will maybe allow them go into the brain, maybe also direct them to specific cell types. So there is this directions that they go to. But what cells then take them up? Are they being taken up by neurons, by astrocytes, by microglia? And when they are taken up, do they have something that modulates the gene regulatory program or the function of the cell? If we can capture this, then we can know what is that beneficial molecule. So eventually, we're going to be able to follow these EVs after exercise, see where they're going, what cells take them up, and how they're influencing the transcriptome of those specific cells. And then perhaps we'll have this full integration of spatial transcriptomics as well, so that you'll see where the EVs are distributed once they migrate through the body. And then we can try to harness that in different ways for people who don't have the same ability to be active, or people with reduced mobility in other diseases, for example. So if we can induce that change without having them be active to start, this would be one of those ideal situations. Another really exciting area is understanding how nanoplastics are infiltrating our biological system. There is certainly evidence that we're accumulating nanoplastics in different peripheral organs, but one of the places that has shocked and surprised people is that these nanoplastics seem to be getting to our brain. So we're going to put nanoplastics in the brain of rodents, and we're going to see where they end up within a cell and how they're influencing the transcriptome. Our perspective is to look at it from a psychiatric point of view, but I'm also collaborating with others who look at whether or not this is influencing the rate of Alzheimer's and Parkinson's and other neurological disorders. We are at the start of spatial transcriptomics, and this community that we've built at McGill is helping each other. So for example, we were very excited to get Loïc Binan and his expertise in spatial transcriptomic, where he's an innovator developing some of the techniques himself. We can use commercial and non-commercial spatial technologies and push the edge of the envelope of what these spatial technologies can do. I work with Joanne Stratton preparing great samples so that we get really high quality samples. The other aspect is that we end up generating just such a massive amount of data. And I may have ideas of how I want to explore the data, but may not have all the tools. That's when I collaborate with people from computer science, and they're all brought into this single cell community to leverage machine learning and AI to really get the most out of them. It requires expertise from all these different fields. For example, one of the more recent publications really worked a lot with an expert, UAD. He's actually established some frameworks for single cell analysis. We learned the sequence structure in individual cells so that we could predict whether or not mutations to the genome are influencing that DNA structure, and then how that's related to the depressive phenotype. And that gave us a lot of insight into how what we get from GWAS can be functional. But without his expertise in using our data to model the sequence structure, we would have never been able to do that. This is really a group initiative that has been supported. And the fact that we're bringing in more talent is only going to make the community grow in a more important way. And I hope it will keep us as leaders in this field. I like doing these data-driven approaches where you can have hypothesis and hypothesis-directed science, but sometimes when it's all new frontiers, let us see where the data brings us. I think this is very exciting times for that.